Hello students, in the previous class we have discussed about the sexual reproduction organism, all the events like pollination the pollinating agents. Today we will discuss about the fertilization. After pollination the second stage is fertilization. So this topic today's session is about fertilization, pre-fertilization and post-fertilization events. Then all the topics that we'll cover today is what are the factors which decide the fertilization, like pollen pistil interaction, stigmatic fluid, then entry of pollen tube by various ways, and like porogamy, salazogamy, mesogamy, then the role of synergid cells, double fertilization, triple fusion, post-fertilization events which would result in formation of endosperm and the embryo. So let's start with the first topic, which is the pollen pistil interaction. Pollen pistil interaction is after when the pollination process occurs, or pollination, which is the transference of pollen grains from male gametophyte to the stigma. So pollen on stigma. Now it is the role of stigma to decide or to accept that pollen grain. So the stigma is having a ability to detect that these pollen grains of its are of its own species, whether to accept or not. So that interaction or that ability of stigma is termed as pollen pistil interaction. Stigma is having the ability to detect the pollen, the viable pollen, or the pollen of its own species. Next to this is stigmatic fluid. Stigmatic fluid, the fluid or the liquid present in the stigma. That liquid present stigmatic fluid is it contains water, lipids, resins, all these some sugary substances. Now the entry of pollen tube or the germination of pollen, it all depends upon this fluid. When the pollen tube germinates, in the pollen tube an enzyme is present. That enzyme, cutinase, just mark a star mark here, cutinase enzyme present in the pollen tube, help in the entry of pollen tube through the stigmatic papilla. So entry of pollen present in the pollen tube. This is the enzyme which is present, and also in the it helps in the dissemination or the process of release of the nucleus inside that ovule. So this, then next is the entry route of pollen. Like this is the ovule covered with the integuments that we have studied yesterday. So ovule, which contains this antipodal cells, the polar nuclei or the egg cell and the synergid cell. Entry route is if the pollen tube is entering through the micropylar end or the shell end. So when the pollen tube is entering through the micropylar end, micropyle is the term porogamy is given to it. Porogamy, entry route of pollen tube. Number one is porogamy. Number 2, chelazogamy. Number 3 is mesogamy. Now here, porogamy, if the entry of pollen tube is through the micropylar end, which is very common method in most of the species. The method is common method of entry or common process in most of the species of flowers. Then chelazogamy, if the entry is through chelazal end, which is in case of example for this is betul. Then mesogamy is the entry route through the integuments, like in case of cucurbits. Next to this is role of synergids. All these are the entry routes of pollen tube. Next is role of synergids. Synergids basically they are not having a main role or function in the fertilization process. They decide the entry of pollen tube, entry route and synergids decide the entry route. Of pollen tube and also the dissemination of pollen tube nucleus. These two functions are performed by the synergids. Next to this is double fertilization and triple fusion. Double fertilization Now the term, this is very important. Frequently asked question, double fertilization and triple fusion. In case of ovule, this is the antipodal cell, the polar nuclei, then the synergid, and this one is the egg cell. Now in this, when the pollen tube enters, the fertilization process is the first fertilization which is with the egg cell and the pollen tube nucleus. Second fertilization is polar nuclei with the pollen tube nucleus. One more thing, in the pollen, in most of the flowers, the pollen tube contains three nucleus. The pollens are shed at 3 nucleus stage, but when they are shed at 2 cell, mostly at 2 cells, not 3, 2 cell stage. Pollen grains. One is the generative nucleus. And the other one is the generative and which one? This is the endospermic nucleus. Oh sorry, cytoplasmic nucleus. These two nucleus, 2 cell stage. When they are shed at 2 cell stage, they are one of the nucleus, generative nucleus, they divide to form the one more, and that they form 3 nucleus in the pollen tube. Out of the 3, 1 degenerates. So here fertilization process, egg cell and pollen tube, this one polar nuclei and pollen tube. Then since this process is it involves 2 fertilization events, so the name is given double fertilization. Then the second term is triple fusion. Triple fusion. Triple fusion. Here. Triple fusion. One nucleus here with the egg cell, two polar nuclei here. This division was the fertilization here. In this one, when the male nucleus which is the N divides with this 2N, it forms 3N stage, which is the triploid. So that the formation of three nuclear stages there. That's why the term is given triple fusion. Two polar nuclei fuses with one male nuclei of pollen tube. Then next to this is post-fertilization event. Post-fertilization event is after fertilization, zygote is formed. Zygote is formed, and this zygote requires nourishment which is given by primary endosperm nucleus. Primary endosperm nucleus, the division or endosperm nucleus, endosperm is nutritive in function. Nutritive in function, it is of the formation of endosperm is of in three ways. One is the nuclear, second is cellular, and third is halobial. Nuclear division, nuclear division, if the division of nucleus, the first division of nucleus is not followed by the cell wall formation, only the nuclear division continues, then it forms so many free nucleus in the cytoplasm and in the egg cell or the egg capsule, and that free nuclei, like in case of Coccus nucifera, which is the coconut, the watery substance contain free nucleus termed as liquid syncytium, which is having nutritive properties, and it can be used as medium for the tissue culture. Next to this is cellular endosperm. In this, if the nuclear division is followed by the wall formation, then it is nucleus and cell wall both are present. So cellular. It takes place basically in the gamopetalous. Gamopetalous means where the petals are fused. Gamopetalous dicotyledonous plants. So this cellular have this one. Then halobial. Halobial is the intermediate between intermediate among nuclear and halobial. In nuclear and cellular, because in this first wall formation with accompanied by nuclear division, forming micropylar and and shellazal chamber. After the transverse division, which results in micropollen and shellazal chamber, free nucleus is formed. Only the nuclear division takes place and free nucleus is formed. So that stage is halobial. Nuclear division, which is later on. First it is cellular division, forming two chambers, micropollen and shellazal chambers. After that, nuclear division only. Next is embryo. After this, the zygote, it utilizes the endosperm as a nutritive tissue and forms the embryo, which is the process is termed as embryogenesis. Endosperm in some like pea, groundnut. These are the cases where the entire endosperm is consumed. Endosperm is consumed in case of pea and groundnuts, and most of the seeds, this endosperm is utilized in the embryo formation. So in the mature stage, endosperm is absent. But in some seeds like cereals, rice, wheat, the endosperms are present after the maturation of seeds also. So in mature seeds, these are the exceptions. Mature seeds, endosperm present. Exception. This is all about the fertilization event and some of the post-fertilization. In the next term or next session, we will discuss about the growth or development of embryo in the dicotyledonous and monocotyledonous structures, then parthenocarpy, apomixis, or few terms. Thank you for today's session.